Hey, what's up everybody, and welcome back to the Sanctuary. I'm your host, Professor C, and we're going to talk some more anatomy and physiology today. Specifically, I'm going to get into the concept of active transport. So, without anything else to do, let's do it. Okay, we just finished in the last lecture looking at passive mechanisms. Just to remind you very quickly, if it was a passive mechanism, substances went from high, right, down their gradient from high to low, and no ATP was required. That was really the only two characteristics that helped us easily spot some sort of passive mechanism. Here, with active transport, we have much ATP required, and it's usually quite a bit depending on what we're doing. But another good thing to think of when you think about active transport is you must use a carrier protein. Now, we saw a carrier protein being used in passive mechanism and facilitated transport, but we didn't need to power it with ATP. Now we do. If you're doing active transport, substances are going to where they're already abundant, and we're pushing more in, right? We're going from low to high. Let me erase my thing there so you can see it. So from low to high, putting some more in to where it's already full, perhaps, and this will require ATP. The classic example, the one you find everywhere, is the sodium-potassium pump. I see very quickly, just by looking at the picture, I'm circling it here, ATP. So we know we're in active world. Right, we know we're in active transport because ATP is being used. It's hard to kind of see here, low to high, so I've made a little notes for us. Sodium is abundant outside of the cell, and look down here at the bottom, potassium is abundant inside the cell. So if the goal here is to pump sodium this way, it's going from where it's a low concentration into a high concentration. We're putting it to where it's already abundant. We're pumping more in. Same thing with potassium here. We're going to pump it in to the cell here. And this is where it's already abundant. So it's going from low to high as well. So not only do we see two different particles here, ions, going from low to high, but we also see ATP being used to make these reactions occur. So this is found, as it says over on the left, in all cell membranes. This is how we're going to regulate ionic concentration in and around cell membranes. And this will be extremely important later when we start talking about how to make muscles fire or how to make parts of the nervous system create electrical impulses. We'll have everything to do with these types of pumps. And so here it says maintains electrochemical gradients for optimal functions of muscles and nervous tissue. That's the fancier way of saying what I just said to you. Be critically important in running these systems. The rule usually is, on tests, 3 sodium out for 2 potassium in, 3 for 2, 3 sodium out for 2 potassium in, ATP is required. Okay, this is different than true active transport. This is called vesicular transport. And the point is we're going to use vesicles. And you can see some of these. Here's a vesicle, here's a vesicle, here's a vesicle, here's a vesicle. They look like balls filled with some sort of product. Think of them as vessels, like ocean ships, delivering crates of material, across the ocean. This is a very similar idea. Instead of calling them little boats or ships, we're going to call them vesicles. So this is a way of moving volumes which are way too large for just pumping across. So we're going to have to use vesicles to transport in and out of a cell. Lots of ATP is going to be required for this. We're going to have to actually physically break the membrane and rebuild it, and that's going to require a massive amount of ATP. Exocytosis and endocytosis are shades of vesicular transport. And I think it's going to give it away here, but exo refers to taking something outside of the cell, vesicles, moving them outside of the cell. And endocytosis is referring to bringing in large packages, I've said here, vesicles, into the cell. So let's look at the two shades here. Exocytosis, as I just mentioned, exo meaning like exit, to leave the cell. And here we see a really great picture of a neurotransmitter being formed in the cell and being pushed out of it in vesicles. Notice how the vesicles will collect this material and will deposit it, push it out of the cell. So secretion is a great term for what you see there. Could be neurotransmitter. This to me looks very much like a neurotransmitter. Could be mucus being secreted. It could be waste being ejected from a cell. But the point is we have large vesicles carrying the product, and they're going to release it from the cell. This is exocytosis. Endo is the opposite. Endo is pulling something into a cell. And there are 3 ways to do this according to this talk. The first is called phagocytosis. Now, phago, anytime you see phage, phago, it's referring to eating. So phagocytosis literally means like a cell eating process. So what you see is it kind of looks like a meatball. It always does in the figures. But what's happening here is the cell looks like it's reaching out some arms, feet, to grab it. They're not real feet, of course. There would be false feet. And you see the term there for the false feet, pseudopodia. So pseudo referring to false or fake. Pod referring to a foot. Think about a pedestrian using his feet, or a podiatrist, a foot doctor, right? So pseudopodia would be false feet. So you're seeing these false feet reach, I think they should call them false arms, but who am I? False feet reach out, grab this meatball and pull it in. So that's what it's saying. It's grabbing solids, eating. That's why we use the word phagocytosis, cell eating. So a form of endocytosis that involves pulling in solid material, cell eating, phagocytosis. Well, what about cell drinking? What if it's pulling in fluid packets? Well, that's the next one. Pino, almost like vino, like it's drinking the wine, right? It's not drinking wine here. It could be drinking just stuff that's found in the extracellular fluid. It's just taking a gulp of it. Or it could be something that was delivered, and the cell now wants to take in a bunch of it because it's surrounding the cell, and this is the only way in. So pinocytosis, referring to drinking, cell drinking. There you don't see any false feet reaching out and grabbing it. It's just like it's taking a big old gulp of the water around it, called the ECF, pino. So phago is eating, pino is drinking. And the other one here is kind of a giveaway if we look at these little teeny tiny Y-shaped structures that I've drawn in several of my pictures. These are receptors, and they're found on the surface of cells. And so this is called receptor-mediated endocytosis. Endocytosis that requires some sort of receptor to help mediate the process, right? So some sort of specific molecule, could be a hormone, could be a neurotransmitter, could be all kinds of different things, binds with a receptor, and this triggers the endocytotic process. So 3 types of bringing in materials into a cell: phago, eating something solid, phagocytosis; pino, drinking, pinocytosis; and receptor-mediated endocytosis, which requires some sort of specific binding to trigger the endocytosis. By the way, on standardized tests, this is the most common form of endocytosis. Thanks for watching the video. If you liked it, check out some more. See you for the next one. Bye-bye.